Welcome you in bio class. I am going to explain you mechanism of enzyme action. You have to know what is enzyme and how is the structure of molecules of enzyme. Enzymes are mostly globular protein. Enzymes have primary, secondary, tertiary and quaternary structure. Three-dimensional structure of protein provide shape the enzyme and form active site of the enzyme. The substrate molecules attach to the active site of enzyme molecules. The active site is made up of a few amino acids that are close to each other. In tertiary structure, when we are studying the tertiary structure of molecule of enzyme, in that active site is made up of amino acids and that amino acids are very close to each other. Mechanism of enzyme action we can study with the help of two theories. So there are two theories to explain the mechanism of enzyme action. First theory is lock and key hypothesis and second theory is induced fit hypothesis. Lock and key hypothesis was proposed by Emil Fischer in 1894. According to this theory, enzyme and substrate molecules have complementary shape. The specific substrate molecules are bound to a specific region of the enzyme molecules. The specific region of enzyme molecules where the substrate molecules can attach, that is called active site. Enzyme react with the substrate molecules and form intermediate complex. So enzyme molecules is here, it react with, this is the enzyme molecules, it reacts with substrate molecules. Substrate molecules are going to attach to the active site. So this is the active site and their molecules of substrate will attach. Then it will form intermediate complex. And this intermediate complex will break again. Then molecules of enzymes are substrate and products are released. So this is the lock and key hypothesis. You see, a particular lock we can open only by a particular enzymes. Like that, a particular substrate is going to react with a particular enzymes. As lock is opened by a key and on the key, when we see the structure of the key, in that we can see there is the active site, that is the notch, and the notch portion of the key is equivalent to active site. So there are active site is the notch portion of the key, and this key can unlock the lock. So like that, molecules of enzyme and substrate, they can attach. So lock and key model account for the enzyme specificity. A particular enzyme only catalyzes a particular substrate. For example, pepsin enzyme can catalyze the hydrolysis of protein. It cannot catalyze the hydrolysis of polysaccharides or fat. So like this, a key can open a particular lock, and like that you see the molecular substrate can fit correctly into a specific active site of enzymes. So this we can explain with the help of lock and key hypothesis. This hypothesis was proposed by Emil Fischer, in that key is equivalent to the enzyme and lock is equivalent to the molecule of the substrate. As key can unlock a particular lock, like that enzyme can catalyze a particular substrate. There is one more hypothesis that is called induced fit hypothesis. This hypothesis was proposed by Dan Osland in 1966. According to this hypothesis, active site of enzyme does not initially exist in a shape that is complementary to the substrate, but it is induced to assume the complementary shape according to the substrate molecules. So therefore, it is called induced fit. This model is also called a glove and hand, and by the Monod and his co-workers, there is allosteric model. In the allosteric model, each molecule of enzyme has two or more binding sites which interact with each other. So these are the two theories to explain the mechanism of enzyme action. First theory is the lock and key hypothesis, and second theory is the induced fit hypothesis.